Okay, good afternoon, everyone. Thanks for joining in a little earlier. For those who were not able to make it to the earlier session, it's being recorded, so you all can follow along when you all view your recording. All right. Just a couple of things before we start with the lecture today. You all would have completed your in-course exam last week. I didn't have any issues that were reported to me. So I'm thinking everything should have gone smoothly. I'm in the process of reviewing your answers, and your marks will be up on banner by next week or before next week, right? So you can look out for your in-course 1 marks. In today's session, we're going to be talking about microbial growth, the first part of that. And I have already assigned you all your projects. It's all individual activities. Just a reminder, you all should have at least looked at that section already and have an idea of what your project is about. And you all should have started your work on that project, right? You all have about a month again to hand up your project, about 4 weeks or so roughly. So make sure if you did not look at it as yet, please log on to your eClassroom page and look at your projects. If you all would remember, it's at the top of your eClassroom page here in this block, your guide. All of the information is here. The due date is the 16th of November. So as I said, you'll have about a month again to hand it in. There's a rubric there for the marking of your project, and you upload it in that section right there as well. So if you haven't looked at your project as yet, at least go on eClassroom, take a look, take a read, and start thinking about ideas for completion of your project. It's due on the 16th of November. It's individual activities for that project, right? So moving ahead, just back to your schedule a bit, we have a live session today. Next week, we have another live session looking at completing this topic on control of microbial growth. And then you'll have 2 recorded sessions. So we wouldn't see each other for 2 weeks. I may ask you all to check in either on the 2nd or the 9th with me just to talk about your in-course exam. So more than likely on the 2nd, we will have just a short 10-minute class or so just to make sure you're on track and just to talk about in-course exam too. So I'll edit that there on your class page, right? So, and then after we have one last live session, which would be on the 16th of November, where you'll have your final class, right? Your projects should be completed by then. So we'll close off the class there, talk about your projects a little bit, review it, and close off class. We should know exam dates and so on by that time. So just a review of your schedule for the rest of the semester. Okay, looking at what we're doing today, control of microbial growth, part 1, your lecture notes are there, which we're going to review, right? And there are some reading resources and video resources. So there's a reading resource, which is really a textbook chapter, and then there are 2 video resources for you all to look at. And on this link here, we have a revision quiz. It's very useful. It has some nice exam type questions. Make sure and look through it, especially closer to in-course 2 for when you're preparing for that, okay? In the link itself, you can go through the multiple choice questions and you can click to review the answers as well. The answers are present on the link itself. Right, any questions for me before we go into today's topic? Good, everything was fine in labs yesterday. I know I told you all the lab lecturers, the lab assistants reached out about just some information, just to remind you all about how it's important to follow your lab rules and stuff. So I hope yesterday was a little better. And I did mention to them your concerns as well. Okay. Right, so we're looking at controlling the growth of microorganisms. So as you should be thinking, when we talk about controlling the growth of microorganisms, we're obviously looking at stopping. How do we stop the growth of microorganisms? You're in the field of nursing, in the medical field, right? You all will be dealing with instances where patients come in with bacterial or viral infections, and your major aim is to control the growth of those microorganisms or to get rid of that infection, okay? So we're going to talk about some factors. In order for us to understand that, we need to first understand what factors affect microbial growth. So what do these microorganisms need to survive? And by removing those things from the environment, we stop them from growing, okay? So we first need to know what they need to grow. So We will look at factors that affect microbial growth. We will also look at encouraging the growth of microorganisms because, as you would know in microbiology, in the lab even itself, when we want to study microorganisms, we need to encourage their growth in a lab setting so that we can look at their structure, their form, we can look at how they react to different environments. And in those ways, that's how pharmaceutical companies are able to create drugs to fight these types of infections or to kill off these microorganisms, okay? And then, of course, from that, so by looking at factors that affect microbial growth, we can provide those organisms with those growth factors or things that they need, environments, nutrients, etc., to grow. And then we can expose them to different types of chemicals to inhibit the growth. And that would be the basis of microbiology and pharmaceuticals with regards to microorganisms and how we control their growth. So as we know with all living things, they require nutrients to sustain life. So if I asked you to write an essay for me on factors that affect microbial growth, you should be able to fully discuss all of these things with some examples, right? And you can talk about simple examples like you go in the lab and you use nutrient agar and you streak a sample from your face, from the ground, from your teeth, from any type of surface, and you put it on nutrient agar, you are sure to get almost positive to get some type of bacterial growth there because nutrient agar, it consists of a wide variety of different nutrients and an ideal environment for bacterial growth, right? So all living things need nutrients to sustain life. The nutrients act as energy, and they can access that energy by tapping into those. Sorry, just have to allow someone in. All right. They can access that energy by tapping into those nutrients, converting them into forms that they can use them, breaking bonds and so on. Water is also essential for life. Microorganisms need water to carry out their metabolic processes. There are certain microbial stages in different microorganisms where they may survive in a dry environment, they may go into dormancy, and then when water is present again, they can continue growing and multiplying. Another major factor affecting microbial growth is temperature. All organisms have an optimum growth temperature, and a common example is us putting food in the fridge. Refrigerators are usually at a temperature around 4 degrees Celsius. That is an ideal temperature to control or stop most type of bacterial growth. So when we put food substances in the fridge, although bacteria is present all around us and it may be present on the surfaces of that food and so on, you're not going to have growth because the temperatures there do not allow for growth of microorganisms. It's not an ideal or optimal temperature for them. So organisms have optimal temperature and pH values where they grow best at. And this is largely determined by the enzymes they have in them. If you all rack your brains a little bit from biology back in CXC A levels, you would have met the term enzymes, right? And enzymes are chemical substances or chemical catalysts. They speed up the rates of reactions. And we know that enzymes work best at specific temperature and pH values. And this is what dictates what temperature and pH all living things would need or require for survival. We have different types of organisms that can be named or grouped according to temperatures that they survive in. Thermophiles are microorganisms growing best at high temperatures. So, for example, organisms you find in desert type regions that are surviving and thriving in those types of regions, those will be considered thermophiles. Mesophiles grow best at moderate temperatures. So, at 37 degrees Celsius or body temperatures, they can grow quite easily once they have nutrients and environments to grow in. Psychrophiles, they prefer colder temperatures like deep ocean water. Psychotrophs are a group of psychrophiles that also like cold temperatures, but more so they're a bit fussy in that they prefer refrigerator temperatures of 4 degrees Celsius. So if you put something in the fridge, like a food item in the fridge, and you put it there and you realize 1 or 2 days you have bacterial growth, it's smelling weird, something is definitely off. And that's not something you're accustomed to because food in the fridge can 4 or 5 days or so, it definitely alerts you to the fact that something is up, right? You are finding possibly that you have psychotrophs present there. I'm just confirming you all are hearing me clearly, right? I was hearing some feedback, so I'm not sure.
Yeah, miss.
Psychoduric organisms, they prefer warm temperatures but can endure very cold or even freezing temperatures. Now, these are some terms that you'll need to familiarize yourselves with. You'll see them popping up, especially so in your multiple choice questions. So make sure and if you all have to take a note of all of those words and learn them off, like make a glossary or something of terms, make sure and do so. It's very useful in microbiology to have a glossary of terms. As I mentioned, pH, whether the environment is acidic or alkaline, microorganisms have a preference, right? Acidophiles, and this one is easy to remember. Acidophiles, they like acidic pH values, a pH of 2 to 5. Alkaliophiles prefer a pH of around 8.5. Another factor would be osmotic pressure and salinity. Osmotic pressure is basically the pressure that is exerted by solutions or more so water inside and outside of the cell. As we know, if we were to define osmosis, osmosis refers to the movement of water from an area of high concentration to an area of low concentration. Or we can define osmosis in the opposite way, which is done in this lecture because it's referring to osmotic pressure. Osmosis can also be defined as the movement of a solvent in the opposite region, in the opposite manner, right? Through a permeable membrane from a lower concentration of solutes to a higher concentration of solutes, right? When the concentration of solutes in the external environment of a cell is greater than that of a solute inside of the cell, the solution is said to be hypertonic. If you all think about the term hyper, when people are hyper, they are very excited. So if we think about a hypertonic solution, it would suggest that there are many solutes dissolved in the water. So it's a hyper kind of situation where you have a lot of solutes present and they are bouncing against each other. They are very high energy. The opposite would be hypotonic. Hypotonic would be where you have the concentration of solutes outside a cell is less than the solutes inside a cell. And that can affect movement of substances, and that can affect how organisms can survive. Organisms need to be in an environment that is ideal for their growth. So osmotic pressure and salinity of that environment can definitely affect them. One example, plasmolysis. With plasmolysis, it's a condition where the cell membrane and cytoplasm of a cell can shrink away from the cell wall. And this occurs when you put bacteria in a hypertonic solution. Reason being, the water is being drenched or pulled out of the organism and the organism is going to shrink. And that obviously is going to affect the functioning of the organism. Less water means less access to that water to break down substances to allow metabolic reactions and for overall survival of the organism. You can also have the opposite happening. If a bacterial cell is placed into a hypotonic solution, meaning you have very little solutes and lots of water, it's a very dilute solution. What you're going to have is water rushing into the bacterial cell, causing it to stretch. And if it does have a cell wall, it's not going to burst. If it only has a cell membrane, it can cause bursting and death of the organism, right? This process is known as plasmotipsis. Then you can have isotonic solutions, which is ideal. An isotonic solution is when you have concentration of solutes in and out of the cell the same. If a cell is placed in an isotonic solution, it means that solution is not going to affect movement of molecules in and out. It's the ideal environment. You have equal exchange or equal movement of substances in and out of the cell. The cell, therefore, is not going to shrink. It's not going to expand. It's not going to be affected by any type of drastic movement of water molecules. So therefore, ideal situation, therefore survival. Some other terms there, halophilic and haloduric organisms. So organisms that prefer salty environments, halophilic. Organisms that can live in salty environments, they like salty environments, but they do not like salty environments, but they can survive. They are considered to be haloduric. And then barometric pressure also affects how an organism survives. Just as we survive in an environment on planet Earth where our atmospheric pressure is stable, we have forces of gravity and so on pulling us to the ground, and it's ideal for human survival, so too microorganisms, they need a specific type of pressure or environment to survive in. There are some special organisms that can survive in very high atmospheric pressures, and they are called piezophiles. This is just a diagrammatic image showing you hypotonic, hypertonic, and isotonic solutions, right? Showing you how you can have shrinkage in hypertonic solutions, bursting in hypotonic solution, and then in the isotonic solution, if you realize the cell is staying the same because there's equal movement. The both arrows show equal movement. There's no difference in the thickness of the arrows. You call that net movement. Another factor affecting microbial growth would be gaseous atmosphere. with gaseous atmosphere, you have organisms needing specific types of gases to survive. And you can categorize organisms. Again, some terms you all need to know. Obligate aerobes would be organisms that prefer the same environment that we do, which is mostly so oxygen. Microaerophiles, they require reduced concentrations of oxygen, about 5%, sorry. Obligate anaerobes, they do not need oxygen at all. They are killed by the presence of oxygen. Capnophiles, they need carbon dioxide, about 5 to 10% of carbon dioxide in the atmosphere for survival. So obviously, if we're culturing bacteria in the laboratory, we need to create an environment that is ideal for them, that has all of these different components readily available to them. Just give me 1 second. Sorry about that, y'all. Right. So culturing organisms in the laboratory, we definitely need to provide them with all of these ideal conditions that we just discussed. A good atmosphere with whatever gases they need, ideal pressure, ideal pH, ideal nutrients that they need for their survival, and so on, okay? And we need to take into consideration the solution, whether it's the concentrations of solutions that they have within their cells, so that we provide a nice isotonic solution for them. So when we think about bacterial growth, do not think about growing in size as we would think about children growing in size into adulthood. With bacteria, when we think about increase in bacteria size, we think about growth in numbers because they are multiplying and forming more and more organisms, okay? So it's an increase in the number of organisms. Bacteria divide by a process called binary fission, and as soon as they reach their optimum size. They continue this process of binary fission through many generations until a colony is produced. Did you all see any colony growth? You all cultured anything in the lab as yet? I can't remember off my head. The labs that you all would have completed so far. Did you all do any cultures as yet? Yeah, no, you all saw a colony of bacteria as yet on an agar plate.
Yeah, miss.
Okay, good. So you all know what a colony looks like, right? When you all did your shrieking, it had nothing on the agar plate. You came back in a week, you saw that growth taking place, those little round colonies growing on the agar plates, right? So that is because of binary fission. It produced a solid culture, a colony on your culture medium. Binary fission will continue for as long as there's sufficient nutrients, water, and space. So if I were to leave that agar plate to grow, it would continue to grow and grow and grow, use out all the nutrients in the agar plate, and then you would see the agar starting to decrease in size. So you'd see the gel shrinking away from the walls of the plate and getting smaller and smaller because the bacteria would be eating it up as a food source. That time it takes for one cell to divide and form two cells is called the generation time. So you'll need to know that definition, generation time. So make sure and take note all of those words in italics, all of those terms in italics. Those are definitions. You all can add to your glossary for revision. This is an image, an electron microscopic image showing you binary fission of Staphylococcus organisms. So when we culture organisms, we need to give them a specific media. In the lab, we are using artificial media known as nutrient agar. With the nutrient agar, we have provided all of the different substances that possibly those organisms need to grow, okay? It is definitely a chemically defined medium because we know what ingredients are in there. We use agar gel. It comes in a powdered form, and the lab techs will have already plated it out for you. When you go into the lab, you just get your plates that already has that jelly-like agar in it, right? They would have used a powdered substance, mixed it with water, and at a specific temperature, it is like a liquid form. And when they pour it into the plate, it gels up and gets that semi-solid nature. All right. So culture media can be liquid or solid. You can have different types of culture medias. If you have organisms that are fastidious, so fussy, and they need specific things to grow, for example, if they need to feed on blood for survival, you would have to make an agar plate that has blood in it. That is called blood agar. Okay. There are many different types of enriched media that you use in microbiology depending on what those organisms need to survive. Then you can have selective medium. Nutrient agar is not a selective medium. Nutrient agar, anything can grow on it. If I want specifically to have one species of organisms growing, and I know that that species of organism, it is very prone to a high concentration of nitrates or something like that, I would use an agar that has high concentrations of nitrates. And I may also put very low concentrations of antibiotics in my plate to prevent growth of other microorganisms while I am still selecting for that organism to grow. So when I go and I plate my bacteria there, and I come back a couple days after, I don't have 10 different types of colonies. I just have one I have selected for that organism by growing it in a selective medium. Okay. You can also have differential medium. Differential medium allows things like color changes. So sometimes the colonies may grow in specific colors, or they may change colors they may cause the agar to change colors in areas where they are eating up the nutrients. Those types of media are called differential media. All right. So you have many different types of media in your textbook. It gives you some good examples of those media types. And I hope I'm reiterating again. I hope you all are reading your textbooks before you come to your lectures, right? Thioglycolate broth is a popular liquid medium that supports growth of all categories of bacteria based on the environment that they need. So if you look at this tube here, this is what a thioglycolate broth tube looks like. It's a tube, cylindrical in shape. And what happens is the broth called thioglycolate broth, it settles in such a way that at each height, you have different concentrations of oxygen present. So at the lowest part of the cylinder, you have 0% oxygen. At the highest part, you have about 20% dissolved oxygen. If I take an inoculating loop and I use antiseptic techniques, flame my loop, sterilize my loop, pick up my organisms, I inoculate this liquid media. So I put in my inoculating loop into there, again using aseptic technique. I swish it around very gently to make sure the organisms dislocate and enter the broth. I take it out. I flame the mouth of my tube to make sure I don't have any bacterial contaminants. I close my tube and I put that standing in a rack to grow for 48 hours. And I come back. I may see cloudy growth around the 5% line in that lower area. And that would tell me that the organism that I had growing there is a microaerophile, meaning it needs about 5% oxygen to grow. If I come back and I see growth cloudy at the bottom only, I know it's anaerobic organisms, organisms that do not like oxygen. If I see growth on the top, obligate aerobes, meaning they need oxygen just like we do. If I see growth throughout the tube, those may be facultative organisms, meaning they can grow in any environment. So that's a nice example of a broth that can be used to show that. So as I was talking about differential media, you're seeing the color changes here that allows differentiation to show that the organisms are present. This is blood agar. Blood agar is considered to be enriched and differential. Enriched, obviously, because it has blood in it. Differential is because you're seeing that color change or that difference in color where you have colonies growing. Okay. We all did inoculation in the lab already. I'm not going to go through this in detail. Inoculation, you all should know how to define it. It's the addition of an organism to a nutrient plate or to a medium using aseptic technique, right? What is aseptic technique? Again, you all did an entire lab on aseptic technique. You all should be able to write about aseptic technique and give examples of, if you go in the lab, what are some things you do to achieve aseptic techniques, right? Flaming your loop, washing your hands, wiping down your counter, making sure your all of your materials have been autoclaved before you use it, all of those things. Incubation, another definition. What is to incubate? To incubate is to leave organisms to grow. Different organisms may have different incubation periods. Generally speaking, for bacteria, we get growth in about 48 hours, right? There may be different types of incubators based on atmosphere, temperature, or moistures. So a refrigerator can also be an incubator if we are looking at a specific temperature the organism needs to grow. If it's environment or atmospheres, there are different incubators that are usually in microbe labs that has carbon dioxide, no carbon dioxide, or no oxygen in it, depending again on if the organism is fastidious. Reminder, fastidious means fussy. All right. Sometimes you may have, in the lab, you need to do a bacterial population count, right? I'm just going to go through this process, this example of how to do a count very briefly. You all don't have to do this. If you all do go into the field of microbiology further, you may come across this is usually more so in the field of engineering, where people need to check like sewage treatment facilities, excuse me, to see the number of microbes that you have to make sure it's not, make sure the sewage is being treated properly. Sometimes people also test drinking water to make sure the microbe count is at values that is okay, that the water is potable water, it can be drunk, can be consumed. Okay, so in those cases, they will do bacterial population counts. Very easy way to count bacteria. You could use a spectrophotometer. It's a piece of equipment in the lab where light is basically shown through a little vial of the substance. And based on the turbidity and the amount of reflection you have in or the reading on that spectrophotometer, it tells you how turbid that sample is, meaning how many bacteria you possibly have floating around in the water. So the more clear it is, the higher your reading on your spectrophotometer. The less clear it is, the lower your reading. Okay. Or you can do something called a viable plate count, where you take a sample of the water, for example, if we're testing to see if it's drinking water. You take a sample, you dilute it a couple times, and then you grow it up on a plate, and you see, based on your volume that you would have put on your plate, so let's say it is 0.001 ml, and you had 159 colonies, based on your charts of comparison saying what is allowed, you can then say, okay, this water is clean or this water is not clean. We need to go and review those techniques of sterilization, etc. Bacteria can be cultured in the lab, and then you can do counts along the way to generate something called a population growth curve. A population growth curve, basically, as it suggests, shows how an organism is growing through time. And this is what a bacterial growth curves look like. You have 4 phases: a lag phase, a logarithmic growth phase, a stationary phase, and a death phase. Now, you all should be able to define and discuss these in detail. On your eClassroom page, you have a nice video resource there about your microbial growth curve. Make sure and look at it, even though we're discussing it briefly here. And there's also really good descriptions in your textbook. If I ask you to write about the 4 phases of bacterial growth, you should be able to appropriately do so, defining each of those phases and explaining in detail what happens, right? The lag phase basically is when the organisms are now introduced to the environment. They're getting familiar with their surroundings. They do not really grow in that phase. It's followed by the logarithmic growth phase, where you have exponential growth. Growth basically skyrockets through time. They use up their nutrients, they multiply, they grow. Then they go through a period of stationary phase, where nutrients are coming to an end. Death rate is equal to growth rate, so you do not have any increases. And then finally, nutrients are no longer present. You have organisms dying off. So you see the curve there starts to go down. Okay. A chemostat is a structure or a piece of equipment in the microbiology lab that you use to have continuous cultures. So if I were, for example, studying E. coli in the lab, and I need to do 100 different tests on these E. coli organisms, that is going to take me about 5 or 6 months to do. I can't be growing E. coli and doing a culture plate every other day and waiting 48 hours for it to grow. That obviously makes no sense. So therefore, a chemostat would be used. In the chemostat, you have your organisms. You are providing your organisms with a controlled amount of nutrients, a controlled environmental atmosphere, temperature, etc., so that the organisms can continue growing in that growth chamber. And anytime you need a sample, you can just go and take a sample and carry out your test on it. Right? The next part of the lecture talks about culturing organisms in the lab. So if we had obligate intracellular pathogens, this would be organisms that need to live inside of other cells. Okay, they need to live inside of host cells. So for example, rickettsias and chlamydias, if someone were growing up these in the lab and they were doing studies on these, they would need embryonated chicken eggs, lab animals, or cell cultures inject them with the organisms, which obviously there's a lot of debate on how humane that technique is. But in order for microbiologists to study these organisms, they must grow them within host cells. Culturing of fungi in the lab, it can be very iffy because fungal spores can very quickly enter the environment and cause contamination. When I was doing my master's in UWI, I remember somebody was growing a fungus, and basically the spores did enter the environment, and a lot of different cultures and other people's work and so on became contaminated because those fungal spores are so easily spread, right? Fungi that cause diseases in humans are also very infectious, so very aseptic techniques care needs to be taken. People need to have proper PPE on and so on when they're culturing those organisms and studying them. Most microbiology labs do not culture protozoa, however, there may be those that needs to be studied that causes severe diseases in humans. Again, there are special labs that are used for those. Again, proper PPE and so on would be needed to do those things. Right. So we spoke about controlling growth, encouraging growth. Now we talk about inhibiting growth, and there are several terms that you'll need to be able to define, right? Sterilization versus disinfection. Sterilization is the complete destruction of all microorganisms, including cells, spores, and viruses. It can be accomplished by dry heat, autoclaving, gases, various chemicals, and other types of radiation. Disinfection is the destruction or removal of pathogens from non-living objects. So when we wipe down our kitchen counters, for example, we are disinfecting, right? You can use physical or chemical methods, for example, pasteurization, another example. Disinfectants are therefore chemical substances that eliminate pathogens on inanimate or non-living objects. Antiseptics are used to disinfect skin and other living tissues. So disinfectants and antiseptics work the same. It's just that you use different terms for living and non-living environments where you are controlling the microbial growth. There are different types of agents that are used to kill or control microorganisms. When we use cidal, so a germicide, a fungicide, we are talking about killing the organisms, okay? So anytime you'll see that suffix cidal, we know that those types of chemicals are going to kill off those microorganisms, right? When we see the term static, not cidal, the suffix static, it means it does not kill the organisms, it inhibits the growth. So you may have static agents that if you use them, they may have a time frame of about 4 to 6 hours in which they will prevent or inhibit growth of organisms, right? So you can have microbistatic agents, bacteriostatic agents, etc. Lyophilization is a common practice in preserving of foods where you have dehydration and freezing. It removes microorganisms from those foods so that they can have a longer shelf life. Sepsis refers to the presence of pathogens in blood. You all should know that term. If a wound is septic, it suggests that organisms are causing an infection. Antisepsis would be methods that are used to prevent that infection. There are different physical methods that can be used to inhibit microbial growth. Heat, you need to take into account the temperature you're using and the time that you are using. It's very important to reach the thermal dead point, meaning the lowest temperature that's killing the organisms in a specified time. There are different types of heat used: dry heat and moist heat. Dry heat, flames or ovens; moist heat, autoclaves. So these are examples of dry heat using a flame or electrical heating device. You all may have seen those heating devices used in like salons and so on to sterilize equipment. And autoclaves would be moist heat, right? It's like a large pressure cooker. And that's what is used in the lab to sterilize a large amount of equipment that is used for your labs. And this is what an autoclave looks like. An autoclave maintains an environment where you have a pressure of 15 psi and a temperature of 121.5 degrees Celsius. Those substances or those materials are left for about 20 minutes and it destroys all living things present there. Usually in the autoclave, they would use this tape. It does not have the black line on them when you put it in. However, when you remove it and you see the black lines on them, it tells you that all conditions were met and all living things should be dead on those different materials. So you can use them effectively without contamination. You can also use indicators in the autoclave. If there's a color change, it also tells you that, yes, you have all your microorganisms killed. Cold can also be used, example of fridge, right? However, it does not kill organisms, it just prevents them from multiplying. Desiccation or drying organisms, they can still remain viable, meaning if they do have access to water again, they can start back multiplying. Radiation or UV lamps, ultrasonic waves, filters, gaseous atmospheres, these are all physical methods of inhibiting growth. Then you can have chemical methods. Chemical disinfection is the use of chemical agents to inhibit pathogens. Disinfectants can be affected whether or not the surface was clean before, whether or not you're dealing with live organic loads, the number one type of microorganism. The concentration of the disinfectant obviously would allow it to be more or less effective. Contact time, very important. So I like to give the example of people cleaning their bathrooms and so on, and they buy these expensive disinfectants and they spray it. And they go and they scrubbing, and nothing happening. They say, oh gosh, I waste my money. However, if you look at the instructions, most of those things say that you need to leave it for 10 to 15 minutes and then remove it from the surfaces because it takes some time for the chemical to work. So contact time is very important, right? Also, the physical nature of what is being disinfected, temperature and pH. So all of those things can affect how antimicrobial procedures, how effective they are, okay? An ideal antimicrobial agent should have a broad spectrum, meaning they kill multiple organisms. It should be fast acting. It should not be affected by presence of organic matter. It should not damage humans. It should leave a residue so that it lasts long after. It should be odorless, inexpensive, soluble so that you can have dilutions that are also effective as well. Okay, so antiseptics are substances used on human tissues. They help to reduce the number of organisms on the skin while not penetrating your pores and hair follicles, so they're not going into your system to affect your body. All right, antiseptic soaps and so on can also be used. And the next two slides just talk about inhibiting growth of pathogens on kitchens and toys. Those are two very controversial things, but since COVID, people's views have changed drastically on those things, right? So you can do a little bit of reading on that. And that would be a little homework assignment for you, right? So from today's session, I want you all to go onto eClassroom, look at those resources that you have, review them properly, and you all can attempt some of those questions as well on the link that I provided. This is the first part of microbial growth, and we will meet again next week to finish off this topic, part 2 of microbial growth. Okay? Thanks again, everyone, for joining in early, and I'll see you all next week for your next class.
Okay, Miss.